What is lipid? Lipid is something that is not soluble in water. So we have studied the structure of lipid. We have done, actually in MD1, we have done the structure of triacylglycerol. We have done this phospholipid. And then we have also done, what else we have done? Cholesterol ester. So we will talk about the digestion of this lipid. We take this lipid in our diet, and we will talk about the digestion. If we talk about the lipid has glycerol. Okay, with this glycerol there is a bond, there is a carbon bond, and with the carbon there is O group. Again there is this glycerol. In triglyceride there is glycerol, again there is carbon bond, and this is O bond, and this is long chain fatty acid. This is also long chain fatty acid, and here also you will see double bond O. So this is what is called ester bond. So this is called ester bond. This is ester bond. So any compound that digest this bond, that actually break this bond, are esterase. So whatever we talk about today is, there will be esterase. Even in your cholesterol, if you see in cholesterol also, there will be esterase. So in cholesterol, if you talk about cholesterol, so in cholesterol also at third position OH group. Here you will see again, if carboxy acid is there, it will be long carboxy acid, and this is ester bond. This is an ester bond. And in phospholipid, there will be at third position, there will be phosphate. So this is ester bond will be there in this compound. Did you understand? So these are ester. So these all will be esterase. All esterase are hydrolase. These are hydrolase because these are digestive enzymes. Let's see this. So in our diet, what do we take? This lipid is very important source of energy. Okay, energy source, and not only energy source, actually lipid is important part of our cell membrane. So this, and it performs in cell membrane, performs very many functions. So including this, that is why this is the significance of lipid. And we take lipid, 95% of the lipid in our diet is this triacylglycerol, and then 5% is this phospholipid and glycerol. And that is why we need three types of enzyme. The enzyme that will digest triacylglycerol, the enzyme that will digest this phospholipid, and enzyme that will digest sterol. Okay, so these three types of lipid, we will require these three types of lipid. Let's see what happens when you take, let's say you have taken pizza, you have taken pizza, and when you take this, when you take pizza, so what will happen? This pizza has cheese inside, and this cheese, you will bite that cheese, and then you will break that cheese into small particle. And then what will happen? The cheese, again, because of your mechanical forces of teeth, it will break into small parts, and then again it will reach where it will reach small intestine. Okay, it will reach, after breaking, it will reach small intestine, and in small intestine, because of this bile salt, it will break into small droplets, and again it will be broken down into a small droplet, and these droplets will be actually covered by bile salts, and this is called micelle formation. So this is how micelle is formed, because of covering of this by bile salts. And then on this bile salt covered droplets, this pancreatic lipase will act. We will see how does it act, how does this pancreatic lipase act on this. So after acting, you can see this is T-shaped structure will be formed. What is this T-shaped structure? We have seen this triglyceride like this. We have seen triglyceride like this, E form. But in this triglyceride, this bond and this outside bond will be broken down by lipase, not this inside bond. And that is why it will form, these two will be released, and it will form a plain, this T type of structure. So remember, this monoacylglycerol will be, this pancreatic lipase will convert this triacylglycerol into monoacylglycerol. And then after this conversion, this monoacylglycerol and this fatty acid will be absorbed. After absorption, you know what happens. This absorption, this will be again reassembled in endoplasmic reticulum into chylomicron. And this chylomicron will be absorbed in lymphatic duct. This is called lacteal, not in blood vessel. Lipid is not absorbed in blood vessel. Remember, you must have been taught. So it is absorbed in lymphatics. This lymphatic is called lacteals. Okay. So this is, you know, this is present in lamina propria. So let's see this. Steps of lipid digestion. So these are the steps of lipid digestion. You can see first step is minor digestion happens in your mouth. It is very minor because in your mouth there is lingual lipase, and very minute amount of digestion also happen in gastric, in your stomach, because they have lingual lipase. These are very minor, they have very minor contribution. Even these enzymes are absent, no problem. So these enzymes have very minor contribution. Now, second phase in duodenum, you know, in duodenum, second part of duodenum, what happens? Duct will open, pancreatic duct will open, and here what will happen? Pancreatic duct along with bile will also join this pancreatic duct, and bile and pancreatic duct will actually open, and there will be what? Pancreatic juice and bile come here, and this bile will cause emulsification of the lipid. Okay, through bile salt, emulsification will happen. And then after what will happen after that? After emulsification, it will be acted upon by these three enzymes. First, this is very important enzyme, pancreatic lipase. And pancreatic lipase require a co-lipase along with it. We will talk about the function of this co-lipase, how co-lipase helps pancreatic lipase. Second important enzyme is cholesterol esterase. And third important enzyme is phospholipase A2. So I told you 2 means this, whenever you see A1, A2, 2 means second carbon, it will digest second ester bond. So you can remember that way, phospholipase A2 will digest second, phospholipase A1 will first. So this is how you have to remember. So this phospholipase A2 will act on second bond. So these three types of bond, remember these three types of bonds are very, three types of enzymes. If you remember, if you understand enzyme, you have already done your what? I have problem because I am doing it in laptop that is given by university the first time, and that is why problem is. So if you understand the enzymes, you will understand digestion. So how does this enzyme act? You can see this. So this pancreatic lipase will act, this I told you, this will convert this E into T, E into T, like this. So this will be eliminated, this fatty acid will be eliminated, E into T, just remember E into T. So monoacylglycerol, this T is monoacylglycerol, and two free fatty acids. So cholesterol ester will break into cholesterol and ester, no problem. And this phospholipase will break the second, you can A2, second bond, this second, this phospholipase is like this is phosphate, and you can see this is second. So this second will be broken down. This is what will result because of phospholipase, this phosphate and this symbol, and then so phosphate group here, a second group is, second carbon is broken, and first chain will be, so lysophospholipid, this is called lysophospholipid, will be result, will result. So remember these three enzymes. If you remember these three enzymes, then everything will be done. Now formation of mixed micelle and passive. So then what will happen? Bile salt will digest, convert into smaller parts. Then lipid will be absorbed in intestine. Okay, and it will be absorbed in intestine. It will be reabsorbed, and then after absorption in intestine, it will be reassembled. into endoplasmic reticulum. After assembly, it will form what? Chylomicron. And chylomicron, you know, chylomicron will be transported. Chylomicron has B48. So these are the enzymes I have already told you. You need to memorize 3 enzymes. First is pancreatic lipase. Second is co-lipase. Pancreatic lipase and co-lipase, they work together. Second is this phospholipase A2. Third is cholesterol esterase. And don't forget this also, because this is very important in neonates. This is very important in neonates. This is actually the source is mammary gland. And these are all are secreted from pancreatic acinar, acini cell. And these two are, you can see, these are the in proform. So pancreatic acini cell, they are secreted from pancreatic acini cell. This is secreted from mammary gland. It is very important for neonates. And I have already told you what is the function of this all. Now cholesterol esterase has very additional function. You can see it will also digest vitamin ester. And it has ability to digest all the bond, all these 3 bond. This, and this, all these 3 bond, ester bond. So this is one characteristic of this cholesterol esterase. And this phospholipase require calcium for its activity. So these bold letters, remember, these are the unique things that you need to remember. You can see action of pancreatic lipase here. So this is missile. You can see you can read from here. So these are all bile salts covered. These are covered by bile salts. These are bile salts. Now these are like, you can see this round co-lipase, and this is lipase. These are bile salt and then lipase and co-lipase. So you can see this monoacyl glycerol here, then this phospholipids will form here, and this triglyceride will take interior position. Cholesterol will also take interior position. Lipase and co-lipase will act like this. So because first this co-lipase will bind with the phospholipid or this bile salt. It will bind with phospholipid bile salt. And then after binding with it, this provide a platform for binding of this lipase, and that is the significance of co-lipase. It is a protein that actually provides the platform for binding of this lipase. And what does this lipase do? Lipase pull this fatty acid, this triacyl glycerol, pull this triacyl glycerol and convert it into this T form, and then to acyl. And this again pull and convert, pull, and this is how this lipase will digest this micelle. Okay, this will digest this micelle into this E into this T, and this glycerol, E into this T. What this T means? Monoacyl glycerol. Remember, this E, I told you from the beginning, E means triacyl glycerol. So you can see it displaces you can read from it, displaces the bile salt molecule to provide binding for pancreatic lipase. This is the function of co-lipase. So pancreatic lipase, it is active in intestinal pH, and then optimum pH is 6 to 8, because in duodenum from acidic it will become alkaline. Up till this jejunum it will be completely become about 8. So it is active in duodenum to jejunum and ileum. So this is the function of lipase. You know, lipase is very easy to understand. You can see lipase here on co-lipase, it actually bind with the co-lipase and digest this, pull and digest, pull and digest, and digest. After digesting, it converts into T, E into T, this E into T, like this. And this T will be absorbed through passive absorption. See, this is a simple thing, because it is lipid, it will be absorbed through a passive absorption in intestinal lipid, in enterocyte. There is passive, no active diffusion, passive absorption of lipid. So let's talk. So lipid digestion is not that difficult. It is very easy. It is very easy. Lipid digestion, just remember these 3 enzymes, done. And this table, just remember these 3 enzymes, this table, cholesterol esterase, 2 important functions, like phospholipase will require this calcium, and this will act on all other things except this triglyceride and this phospholipid. This cholesterol esterase has capacity to act on all the lipid, even in triglyceride, all the ester bond of triglyceride.